Good evening, everyone. Very good evening to you all. Please confirm if you are able to hear me. Good evening, all the attendees. I can see many people who have already joined the link. Good evening, everyone. So, on behalf of entire organizing team of Biotechnica, I, Tithi Saha, academic support expert of Biotechnica, warmly welcome you all to today's exclusive webinar titled on NGS and Multi-Omics Internship Guide, Skills, Tools, and Projects to Learn in 2025. Thank you, Ningthojam. Good evening. Good evening, Yogita. Welcome. So, guys, whether you are currently pursuing your undergraduate or postgraduate studies in life sciences, or you are a researcher looking to expand your skill set in genomics and data driven biology, this web session is thoughtfully designed to help you navigate the fast growing landscape of NGS and multi-omics. So, whether you're planning to take up internships, transition into industry roles, or pursue research careers, you will find actionable insights and practical guidance throughout this session. So, as this field of biotechnology and genomics continues to evolve at a rapid pace, next generation sequencing, which is also popularly known as NGS and multi-omics technologies, have become central pillars in modern biological research. But with so many tools, so many platforms, and so many different learning paths out there, how do you decide where to begin, or what to learn, or how to build a career ready project portfolio? That's exactly what we aim to answer in today's 1 hour session. So, we have brought together three expert speakers who will walk you through every critical aspect of preparing for an NGS and multiomics focused internship, especially with 2025 in mind and the kind of tools and the skill sets which are required in this year. So, here's a quick look at what today's session holds. So, first up, we will have Miss Snigdha, who will lay the groundwork by discussing the core skill sets you need both wet as well as in the computational lab to thrive in this particular domain. She will also share tips on what foundational knowledge you can start building right now during your undergraduation or even post graduation studies. Next, we will hear from our guest speaker, Mr. Yogesh Joshi, who will highlight the key tools and platforms to focus on in 2025, ranging from data analysis tools like FastQC, HiSat2 to scripting with Python and R, and using cloud platforms for hands-on training. Our third speaker, Dr. Ilamati Natarajan, will guide you through examples of real internship projects and that truly stands out. She will also talk about cross-disciplinary ideas from cancer genomics to microbiome profiling and how to document your work effectively using GitHub reports and presentation. Uh, Followed by these three sessions, we'll conclude our session with a live QA session where you will get the chance to interact directly with our speakers. So I request everybody pay attention during their talk and keep your questions ready so that you can ask them during our question answer session. And towards the end of the webinar, I will also share some information about our upcoming internship opportunities and how you can apply and what kind of mentorship and hands-on experience you will be offering. And before we move ahead, here is something exciting for you all. Okay, everybody pay attention. So a little surprise for everyone joining us live today. We have prepared something very special for you. It's a free, completely downloadable resource designed to be genuinely useful as you plan your next steps in NGS and multi-omics space. But here's the catch. This exclusive surprise freebie will be shared only at the end of the webinar. Okay, so trust me, guys, if you are serious about making the most of your internship and research journey, you will want to stay till the end to grab it. Okay, so everybody stay focused, stay with us till the end, and we will be sharing this freebie along with the kind of knowledge that all our speakers are going to share with you. So, once again, I thank you for joining us live, and I encourage you to stay engaged, take notes, and ask your question in the chat box as we move forward. So, without any further ado, let's begin our first with our first speaker. So, our first speaker today is Ms. Snigdha Tiwari, who is highly accomplished senior research fellow from IIT Roorkee with over 5 years of experience in computational biology, bioinformatics, and telemedicine system design. Welcome, ma'am.
Good evening, ma'am. Hi.
Okay, so before ma'am starts the session, let me give you a brief introduction about Ms. Snigdha. So Ms. Snigdha has made significant contributions in the intersection of basic and applied research, particularly in protein and protein ligand interactions, molecular docking, and simulation studies, as well as healthcare and clinical data management. One of the highlights of her work includes leading the development of Utsarjan app, a digital health platform designed for pediatric nephrotic syndrome data collection and clinical research in collaboration with AIMS Delhi. Currently pursuing a PhD in computational biology at the Translational Bioinformatics Lab, Ms. Nigdha brings a rich academic foundation with a background of chemistry and bioinformatics, alongside a passion for interdisciplinary research and advancing biotechnology and healthcare through innovation and collaboration. She currently serves as an academic support specialist in the Bio IT and Clinical Research Department in Biotechnica. So over to you, ma'am. We are very eager to learn about this multi-omics data science and how we can prepare for it.
Good evening, everyone. Thank you so much, Tithi, ma'am. Uh, Hi, guys. Good evening. So today we'll be talking about the foundations, the foundations and the skill set you all need for starting your career, starting your academic journey, the project, picking up any project in the academia, or if anyone listening to me is planning to transition into the industry. So what are the foundations and the basic skill set one needs for for the NGS and multi-omics internships and multi-omics data analysis? So first I'll talk about what is NGS. What is a next gen sequencing? So if I'll talk about NGS, it is the revolutionary technology that enables now for a rapid and high throughput, and of course the cost effective sequencing of DNA and RNA. You must have heard about DNA sequencing, RNA sequencing, whole genome sequencing, and many of them likewise. There was a time when the Sanger sequencing used to happen. All that labeling the fluorescent and with the wet lab experiments, dropping up the ddnps, and all of that long procedures that Sanger sequencing started with. But now as we go along towards the globalization, towards the big datas and machine learning and artificial intelligence, with all of that, things has really transitioned into rapid and high throughput, cost effective sequencing. Be it's the DNA or an RNA sequencing, the gene expressions, any of it. So beyond Sanger, if you talk about, we moved from sequencing one fragment at a time. If someone deep dive into Sanger sequencing, and then relatively things have evolved with the iron pore and then nanopore, now packed biosequencing. So starting with the very old Sanger sequencing, it was the one fragment at a time, to so much time, so much time taking, so much not very cost effective, of course, to now coming up to millions in parallel sequencing, millions and high throughput sequencing now, and which is very cost effective, which we call as the next gen sequencing. So what impact it has made? It transformed biology, medicine, and agriculture. Of course, it has transformed our our healthcare domains, biological domains, medicine domains, even the agricultural domains, as a lot. So and the key applications, if I talk about, what are the basic key applications that that one can learn with next gen sequencing and coming up, every day, high throughput sequencing and all of that is coming, new generational sequencing is coming up. Talking about the key applications, with working with NGS, one can do whole genome sequencing, sequence the whole genome, of a single cell or a DNA or a whole gene of a body. Exome sequencing, the coding, if you talk about the coding ones, the protein expression ones. um, we can do the RNA sequencing as well, gene expression analysis, all of that. uh, Also, epigenomics and chipsec is one of our key applications. If you talk about the interactions of a DNA protein, histone proteins, chromatins, then chipsec and epigenomics for you. Also, metagenomics is one of a kind. Metagenomics deals with the gut microbiota, microbiome, environmental genomics, and all of that. Also, if I talk about single cell sequencing, is also one of the applications. Single cell sequencing, as in the sequencing of a single cell, that small, all the sequencing for for about a single cell. The basic key applications that that multi omics, or we call it as genomics, or the omics is. uh, Talking about the power of multiomics, what is the power of multiomics? The concept of omics actually refers to the comprehensive study of a biological molecule. Say for example, a genomics or proteomics, a metabolomics, all of that. The comprehensive study of any biological molecule is actually called as an omics. So multi-omics is nothing but it is just the integration analysis, the integrated analysis of a multiple omics layer, from the same biological sample. Analysis of a multiple omics, say for in the different domains, like genomics, proteomics, or all of that, is the multiomics and the omics concept. So why multiomics? If we talk about why the genomics, multiomics has evolved, why Sanger sequencing has evolved. High throughput sequencing and cost effective sequencing is the need of an hour. Then because it gives us the holistic view. Individual omics provides the snapshots. Multiomics paints the full dynamic picture of a biological system. So if we want the full dynamics of a picture, the full dynamics of a genome, how it is the DNA and the chromatin is interacting, proteins are interacting, the whole of a holistic view of any of the omics, like genes or proteins, then multiomics comes into the picture. If we want to get the deeper insights, say for example, uncover the complex interactions about anything, like about a gene or a protein or metabolics, any of that. Working nowadays, working up with the gut microbiota, microbiome, metabolomics, and all of that is trending. So it gives you the deeper insight. Say for example, if it is
Excuse me,
uh,
Snigdha ma'am.
yes.
I'm so sorry to interrupt you. Actually, your PPT is not visible, ma'am.
Okay.
Can you just reshare?
Yeah, yes, ma'am. I'll just reshare. Share screen. Okay. Let me know, ma'am, if it is visible.
Yeah, sure. I'll let you know.
Yeah. I hope it is visible, ma'am, now.
Yeah, just
Okay.
yes, please put it on the slideshow mode. Yeah, now you're good
Yeah.
to go. I'm so sorry to
Okay.
interrupt you. Please
Okay.
continue.
No, sorry for the inconvenience, ma'am. Okay. So now I was talking about the power of multiomics and why multiomics. Why do we need a multiomics as the for the deeper insights, and why to know about multiomics? So because it gives you the holistic view, because it gives you a deeper insight, because disease understanding. For all of this, we want a multiomics to to be to work with us, to get the deeper insights, for example, to uncover the complex interactions between genes, proteins, and metabolites. There are different genes, different proteins, and different metabolites, say for example gut microbiota and all of that. We of course, getting ahead with what is genes, how they are interacting, how they are giving you a dynamic picture of all its interaction, of all its expressions. What is working for you, what is the central dogma, how the transcriptomics is happening, how it is transcribing as a protein. So all of that will definitely give us the deeper insights on everything, on beat it as the health outcome, or maybe working with the plant, transcriptomics, genomics, RNA and DNA will give us the agricultural front, how to overcome the difficulties in that domain, all of that. Also, disease understanding, identify biomarkers, understanding disease progression, how the disease is progressing, maybe the oncological diseases, many rare diseases, all of that, predicting drug responses. How can we go ahead with the holistic view of the drug responses in the body. So it gives us the deeper understanding as well, the disease understanding as well. The key omic layer that one can see, that one can can take up as an interest, as an individual interest, is genomics, which will give us the DNA fingerprint. The next one can be the transcriptomics, which will give us the gene expression levels, the RNA transcriptomics. Also, one can work with the proteomics, the protein functional molecules, how protein and the DNA sequences, or the sequences, that is involved, the exome, introns, and exons, all of that, layers and different molecules of a proteomics and proteins, and understanding them deeply. The next thing that one can work with is metabolomics, that is, your metabolites, the real time cellular activities, all the cellular activities that is going ahead with the body, or the metabolomics of it, and all of that. Also, epigenomics is one of a very key omics layer, which deals with the DNA modifications, your gene regulations, the chromatin, how DNA is interacting with proteins, and all of that. Talking about NGS and multi-omics workflow. Now, what main agenda for my interaction is that I want to tell you that how multi-omics workflow takes place, and what is the basis, what will be the fundaments of wet lab experiments, of the dry lab experiments. What can be the fundamental start, and how can you kickstart with all that skills that is required, for NGS and multi-omics workflow, to know how to start with the NGS pipeline, working with genomics, proteomics, or metabolomics data, all of that. So wet lab, starting with a wet lab, that is a sample or a raw data. One can work with the wet lab experimentation, the quality control, how to trim, then differential expression, expression analysis, and all of that. Sample collection, say for example, while working with the genomics, proteomics, or your metabolites, you need samples, right? Any tissue samples, blood samples, blood cells, or maybe you're working with the microbiome. Gut microbiota is one of an example. Any sort of the sample that you need. The nucleic acid extraction, say for example, one working with the DNA, RNA isolations, quality and quantity are crucial. Working with the quality controls and the isolations is one of the another step. Going ahead to prepare a library, of course. The fragmentation of the nucleic acids, fragmenting nucleic acids, then adding adapters to it, that will help you. That will help the primer to add in, and barcodes for sequencing, all of that. Getting ahead sequencing, running samples on NGS platforms. As nowadays, NGS platforms, very cost effective and high throughput platforms like Illumina, Oxford Nanopore, PacBio, all of that, to generate raw sequence reads. So this is a snap for NGS and multi-omics workflow that one can go ahead with the basic pipelines. Now, talking about some basic foundations that one should have in the dry lab experiments, and even for the wet lab experiments, to start or to take up the career, maybe transitioning into the industry or taking up any academic projects or sort of that. Talking about the dry lab, the dry lab inside. Data is very important. First of all, data to insight is is the journey for a dry lab person. Quality control and pre-processing is one of the step that needs to be mastered. Quality control, and then how trimming happens, how the alignment happens with the help of the star aligners. Counting, that is we work with feature counts, as the quantification, and then we work with DSEC too. There are a lot of Python libraries, talking about the dry lab experiments, lot of Python libraries, lot of imports that that can we work with the quality control and alignments, all of that. The whole NGS pipeline can be worked with the dry lab and the wet lab experimentations together for the best approaches. Quality control and pre-processing is filtering low quality reads, trimming the adapters, with the help of the low quality reads, quality control, fast QC, feature counts, star aligners, the ESEC2 imports, and a lot of libraries also is there in Python and in R language as well, where we can do all of that. That is all the basic of the NGS pipeline. The pre-processing is must, when you get your your data for the analysis, for further analysis. um, Talking about the alignment and assembly, mapping reads to the reference genome, or assembling a new genome. Once we do the quality control and pre-processing, like trimming also happens on the different thresholds. Maybe we look at the FRET scores and all of that for trimming, and how the quality is working. And then going ahead, the alignment and assembly, mapping reads to a reference genome, or assembling altogether a new genome. So that we do the alignments, that I'm talking about, with the help of a lot of tools are there, star aligners, HiSat to all of that. Counting reads, quantifying gene expressions, and now we have to we have to quantify the gene expressions, right, the variants. The counting reads, quantifying genes and expression variants, all of that needs to be done when working with the dry lab pipelines. um, so then at the end, once you master all the quality control, pre-processing, and then the alignments, then counting the reads, quantifying the gene expressions, one will move towards the statistical analysis. What's your hypothesis, what's how the hypothesis is working with the samples, what is the p score, what is the set score, your hypothesis is working or not, all of that. One the dry lab person needs to sort out the all the understanding of the statistics, identifying the significant changes. Say for example, differential gene analysis, we do differential gene analysis, DESEC2 as we call, in the dry lab analysis. So it has lot of libraries we have in Python, in R, anything that that you're well versed with. I'll highly encourage the dry lab people to have an insight for the Python and R basic programs and imports. That is very important for pre-processing, for statistical analysis, all of that. After one the researcher has done the statistical analysis, may move to data integrations and interpretations, combining the multiomics data, the pathway analysis, and network biology, of course, the new age machine learning. Once we have a lot of data, one can work on the machine learning pipelines, how to validate, how to test, dividing into the test set, validation sets, what are the error levels, how the error levels can affect your data, the tweakings, all of that. Then visualization, of course, visualizations like creating insightful plots and dashboards. We do it as the heat maps, PCA plots, um, uh, all of that one can do for the visualization. Of course, that is also a dry lab part, which which needs a lot of Python libraries, R libraries, say for example ggplot2, matplotlib, to import and to visualize your data. For PCA plots, we do dimensionality reduction. So these are basic prerequisites that works for the dry lab visualization, pre-processing, and all of that. Talking about key skill set in demand, the key skill set that one needs for picking up the NGS as a project, or as if transitioning into the industry or anything like that. The hybrid professional, as in the wet lab expertise and the dry lab expertise as well. Both is important, as for now we have the big data. We have to do the big data analysis, write a lot of scripts, like for analyzing, for trimming, for quality control and whatnot. For end to end analysis, one needs to know the wet lab experiments and the dry lab as well. So talking about the wet lab expertise, basic molecular biology techniques for sure, like DNA RNA extraction, PCR, cloning. One needs to master that. Then NGS library preparations and sample quality controls. What are the library preparations, how the adapters are made, and then the primers are set up, alignments are made, quality controls are done. Then experimental designs and troubleshooting, the experimental designing, protocol writing, how things will move ahead, and the troubleshooting, everything is the wet lab expertise that one needs for next gen sequencing. Talking about the computational or dry lab expertise, programming is must. Programming, say for example, any programming you pick up, if anyone is well versed with Python, that will work, or the R will work. R is a good language to work with, biological tools, and with NGS R make it really easy. It has a lot of imports, pipelines, visualization tools, all of that. A lot of GitHub libraries. Whoever is starting for new, just go ahead and see a lot of GitHub libraries, and GitHub scripts are available, to for your hands-on, starting your hands-on. Then a command line experimentation, command line analysis, which we call in the dry lab as a bash scripting. So that is one of a very important skill set to develop. Working with the command line, bash scripting, Linux and Unix is very important. These are the basic tools that we learn. Say for example, making a directory, to copying, to moving, how to grep different words, and then replacing them, because these are the basics of bash scripting that everyone must be needing for pre-processing, for your visualization tools, for all of that. Coming towards the bioinformatics tools, this is one of a very important factor that computational and dry lab person have to pick up. These can be in the form of learning different tools that I've already spoken about. A lot of tools that is already available, star aligners, feature counts, HiSat2. These are few of the bioinformatics tools that is available out there for free. These are the open source libraries. These are the open source. We have the imports, the binary setups as well. One can extract the binary setups and install in the desktop or any of the machines that you are using. Then aligners for variant callers and quantifiers. So there are a lot of bioinformatics tools these days. There are different servers as well. There's this open source server called as a Galaxy server. One If anyone is starting their journey with the basic analysis, dry lab analysis, definitely go ahead, use that. That is one of very basic and very nice, to start with. Then statistical and the data management. How to work with your statistics, with your data. You have a sample size, and working with the sample size on the basis of statistics, with that mentality is very important. When you once you get your variations in your data, how do you work with the curves, Is it a one-tailed a data setup, or it's a two-tailed one? Is it falling into a normal curve, a Gaussian curve? How to set up the sample size, hypothesis, alternative hypothesis. These are some of the basic statistics and data management things. For your file formats, your visualization file formats and extensions. This is very basic and very important, actually. Very important computational and dry lab expertise that one should have with starting with any of the NGS tools like genomics, proteomics, or any of that. Cloud computing in HPC is is the added the important one, I would say, because cloud computing uh, is one of very important computational and dry lab expertise. How to work with your high performance computing, what are the nodes, what are the threads, how the threads and nodes are working, what are the command lines that one should go ahead with, working with all of these HPCs. There are a lot of containers that we put in, s- uh, process managers, crash cron jobs, crash lytics. HPCs and cloud computings is becoming one of a very important hands-on while working with the large setup. Guys, now we are working with such a large setup of genomics and proteomics, big data analysis that we call. One should have that expertise for sure. How to prepare during UG and PG years. So whoever is listening, me, preparing around their UGs or PGs, guys, build a strong foundation first. Go ahead, read about all the the dry labs, how how the languages work, how cloud computing works, how your basic NGS pipeline works, actually, with the with the basic open access tools, bioinformatics tools, imports, GitHub libraries, all of that. That is that is all available. For academic, major and minor in biology or CS or statistics or bioinformatics, may go ahead and start their careers in NGS. Talking about some of few of the core courses that is available is molecular biology, genetics, biostatistics, any of the computer science, programming, algorithms. Definitely can move ahead in the field. Talking about computational perspective, then practice coding daily. Learn your bash scripting, learn your Linux command lines, learn your version controls, like like how to maintain your GitHub library, how to import, how to all of that. And then participate in hackathons is one of an added way of uh, elaborating your expertise. Participating in hackathon, listening all that that is that is available, the webinars and the conferences and everything, um, is one of a very crucial part. So gain crucial hands-on experience, whatever is the hands-on experience, whether it's a wet lab, whether it's a dry lab, gain that, because you have a lot of time, your 2Gs and PGs. Go ahead and do that. Then research projects, one can pick up a projects based on NGS Multi-Omics lab, a wet lab or a dry lab exposure. And from there you can build your resumes, post it on GitHubs, and get started. Internships, different internships for you, explore opportunities in academia, biotech, pharma, hospitals. Now is the time where the NGS is exploding. Everyone everywhere wants, be it's in the academia or biotech industries or pharma, or even in the hospitals, people wants your expertise with computational or wet lab experimentations to go ahead with all the analysis, whether it's a genomic ones or a proteomic ones, metabolomics ones, any of that sort. Workshops, attend local workshops, online sessions, NGS data analysis, R in Python, bash scripting, cloud computing for bioinformatics is a must. Continuous learning and networking, guys, read scientific literatures, which all that are available, stay updated with the new technologies and findings, attend seminars, webinars and conferences, learn from experts, listen to them, network with your peers, join different communities. There are a lot of communities out there for bioinformatics forums, Stack Overflow, GitHub, BioStars. There are a lot of them. You can join, participate in the discussions, learn a lot. Also, working out with your errors. That communities help you a lot. While discussing and learning a lot. Exploring with your errors, working out with wherever you're stuck, building a portfolio, like showcase your project on a GitHub repository or a personal website, any of that. Talking about at the end about the key takeaways is the interdisciplinarity is a key now, because things are interdisciplinary. The wet lab experiment, going ahead with any of a if any of a kind, starting from a wet lab, cell and cultures, towards the computational biology and machine learning, bridging biology, computation, and statistics is a key. Hands-on expertise is definitely will work ahead at any time of a career, any time of a transitioning career, or in the academy as well. Apply what you learn through projects and internships. The field is evolving rapidly, so never stop learning, keep listening and learning. Hands-on practice is a must. Passion and curiosity for sure, drive your explorations of life's molecular mysteries, and keep your passion and curiosity alive, and keep learning, guys. That's all from me. You're not successful, the word says you're successful when you say so. So today, say that. Thank you so much. um, Giving it over to Tithi ma'am.
Thank you so much, ma'am. What a wonderful session that you have presented. Thank you so much, ma'am.
Thank you, ma'am. Thank you.
So I must say that especially the way you broke down the foundational knowledge and the skill sets which are needed in NGS and multi-omics, that was incredibly helpful. And I'm sure all the attendees here truly agree with me, right, everyone? And you also get to know that how to create the right balance between wet lab techniques and computational tools, isn't it? So as I was saying that I'm sure that many of our attendees are now feeling more confident about how they can start preparing during their graduation and post graduation years. And thank you once again, ma'am, for setting the perfect foundation for today's discussion.
Thank you so much, ma'am.
All right, guys. So now it's time to move into our next segment of our session, one that focuses on essential tools and platforms you must get hands on with if you are aiming for an impactful internship in NGS and multiomics data analysis. I'm delighted to introduce our next speaker, Mr. Yogesh Joshi, a seasoned professional who brings with him over a decade of combined experience across academia and industry. Thank you, sir, for joining us and welcome to this session.
Hi, hi. Thank you, everyone.
So, sir, we are truly eager to know, to listen to your talk and the kind of expertise that you bring to us. But let me introduce you to all our attendees today. All our students, Mr. Joshi spent 5 years as an assistant professor, where he guided and mentored students in areas such as genomics, bioinformatics, and computer-aided drug design, sparking curiosity and innovation among young researchers. In the industry space, he has worked extensively on NGS data analysis, pharmacogenomics based testing, genetic test report development using Python and other powerful tools. His expertise also extends to molecular modeling, virtual screening, and identifying novel protein targets for therapeutic applications. What truly sets him apart is his commitment to solving complex biological problems using data driven approaches with a focus on precision medicine and real world clinical impacts. So, if you're wondering what tools to master, which platforms are most relevant in 2025, and definitely how to build a pipeline or script for that matter, so you are in for a very practical and eye opening talk. So, please join me in welcoming Mr. Yogesh Joshi. Over to you, sir, and we are
Yeah,
extremely eager and looking forward to your talk.
yeah. Thank you, Tithya, for giving me this opportunity. Yeah, can you just give me my screen is visible?
Yes, sir, it's visible. Please go ahead.
Yeah, yeah. Thank you, everyone. Very good evening, everyone. I'm Yogesh Joshi, as an bioinformatician, NGS expert, as an engineering lead in the domain of bioinformatics and data science. So today, I'm very excited to walk you through the essential tools and the different biological and the computational platforms which are shaping and the future for to making the biological data analysis in the 2025. So the session is the tools and platforms which you have to must learn in the 2005. See, this particular, I'm just getting this subtitle, which make the empowering the next generation of bioinformatician. So in this particular walkthrough, we are going to detail understand about how this NGS data has to be handled and as an bioinformatician. So whether you are the BSc, master, researcher students, or any professionals also. So this session, it will be help you to understand what's relevant with the industry and what we have to cover from the basics from the academics and what's going on in the industry as in trending and how to get started. So yeah, let me start with this the presentations to understand about the agenda of the my discussion. Yeah. So here I'll try to cover today. First, we will look what are the essential tools which use in the omics data analysis. And later we will explore some cloud platforms and which are the open access resources that makes the data has to be analysis more accessible way. And also I'll share some practical tips to on learning this data pipeline and how to run the scripting. So definitely we will cover some open source and the commercial tools which are available in the bioinformatics. And finally, we will have some discussion on that. Most of the relevance we are starting to the internships, trainings, and the students who are entering in this field. Yes. So let me start with the first. Yeah, why the tools and platform is matter, because before these presentations Nita has discussed about how the dry lab and wet lab has to be combined to solve the biological problems. So I'm not going in detail like how to sample has to be prepared up to the data. So we are more focused on as a bioinformatician part. So yes, it is obviously we need to understand when we are working on the dry lab, like how the data has to be started, what is the sequencing technology, right? So this is what we need to understand from the basics. Yes, that is the what basics of biology. Yes, now it is a completely trending in the that is the next generation sequencing in the biotechnology, in the bioinformatics, or if you go in the clinical and diagnostic setups also. So the day by day data is um, explosively increasing. If you see the graph, so before I think everyone, if you are belong with the some bioinformatics or the some tool, some concepts which you have implemented in your the subjects like NCBI, we know. So if you see the growth rate of the sequencing from the NCBI after the 1990, how the technology has been increasing, similarly how the NGS has been increasing, it is exponentially increases. So people, the researchers, they are initially they are more restrict themselves in the part of research only the gene and protein. Now everyone is focused on the genome. So that is what like because it is a very cost effective technology, right? So everyone we are working on the genomics and different types of the omics. So we are getting the data, but how this data has to be handled, we require a lot of computational the power to understand this data, to store this data, to work on this data. So that is the first what explosion which we have stated. Like before we dive into these tools, we need quite to why they particularly these matters is having to understand today's research environment, the explosion of the omics data. Yeah. So in the last decade, we have seen there is the explosion of this omics data. It may be genomics, proteomics, transcriptomics, and metabolomics. So like the technologies, the next generation sequencing and the single cell analysis, they are generating the terabytes of data. The data is to be TB of data. It is not. Initially, we see there is the KB, MB, GB, but now this biological data is in TB data. So to handle of this large scale of this data, we require a very scalable and the flexible tools. So once we know about this, definitely we can process, we can analyze and visualize the data very efficiently. Next, we will more focus on the tools. So these tools, which we can say as a students, we are learn a lot of theory, right? So we know about how the central dogma of molecular biology is working, what is the gene expression, what is the biological different pathway, metabolomic pathway, and the the different concepts of the molecular biology. But if you have the application, the Python, R, so along with that, which are the different library which use in the Python and R? For example, we can say the Seurat, the cytoscope, and the Python scripts, how to work on this. So these particularly the applications of this programming, it help you to apply that we can solve the theoretical data to into the real world data sets. So this is what they are bridging between the classroom learning and the actual research. So the third, we have to select the choosing the right platform when you are working for the understanding the efficient analysis and to working on the reproducible research. So the platform what we are choose, for example, if you say the Galaxy, or you can say any platform like the R Studio, or you can take any other the black box, the NGS tools, or the cloud-based solutions. So these particularly platforms, they are impacted on your workflows. Whether the workflows is your WGS, WES, RNSX, metagenome, whatever we are working on any workflow. So we have to select a very perfect, right platform to make to reproducible. So a good platform, it ensure your analysis is very efficiently, meaning it saves our time, it saves our resources, and it also helps with the reproducibility. So which is very critical in the sciences. So somebody has to be helped or somebody has to be reproduce their results using same pipeline. Means we are creating some data pipelines so others will be used. This is the current trends, what is going on. So that is what like it is this research it becomes more incredible. So transition of this, what we need to select the students, researchers here, we need to understand uh, what are the different essential tools which we can start to learn in this 2025. Yeah, we will back on this omic data. I think Snigdha has covered in detail understanding how this omics data is important in the life sciences, in the bioinformatic researches. See, this data now it is called as the big data, right? So we heard the term big data, right? So that is not a different. If you see the single sequence from the protein of the protein database or the gene sequence from the gene database, right? So similarly, you can handle the genome data, right? So this is what you can consider. This is the genome. If you see in the UniProt, you are getting the proteome data. For example, mycobacterium tuberculosis, you can download all the different mycobacterium proteomics sets. So this omic data, it encompasses a wide range of the biological informations. It may be DNA, protein, metabolite, the environmental DNA, what are the different types of the phenotypes and RNA. So which makes a system level understanding of the lives, right? We know about the DNA, which is more focused on the genomics. So genomics, which is more focuses to understand the complete DNA sequences. Along with that, to understand how the genetic makeup of an organisms. So which enable this genomics part, it enable to get insights into the gene along with its structure and its variation studies. Next, we will have the transcriptome analysis. So most of the terms which we are called, that is the RNSeq analysis, right? So this particularly omics, it has to be examined how the expression has to be understood and what is the pattern under the different types of the condition, for example, normal in the tumor, right? Next, the proteomics. We know about the proteins. Proteins are the dynamics, right? Proteins are the dynamic biomolecules. To understand these proteins, we need to analyze the full set of proteins. What? Along with their modifications, we need to understand how they are interacting, that is the protein-protein interactions, how they are interacting with the other biomolecules, so which reveals it will give the functional outcome of the gene expression studies. Next, we will have the metabolomics, which include the metabolites. So we have to study the small molecules and the metabolites. So which offer a very snapshot how this cellular metabolic activity is working in the living cells. Next, we have the metagenomics. So this particularly genomics, metagenomics, it give insights, it investigate how the genetic material they are recovered directly from the environmental samples, right? So we need to understand how they are useful to study the microbial community. And later we have the phenomics, which we have the phenotypes. So this particularly omics, it has to be observed what are the different triads and how they are working on the phenotypes. It is linking to the molecular data and linking to the physical characteristics along with the responses. So these omics, they are different layers. They form the foundation for to understanding the integrative and the personalized biology. So this what this is the complete omics data, what we need to understand to work on this cloud. Now I'll back on these cloud platforms and open access resources. So students, as an students, or like if you are working or if you want to work in the bioinformatics, see, initial era was not there is the single sequence, single, there are the single genome will be there, but now data is working. We are not restrict ourselves onto the laptop and our systems on the small space of the memory. We have to move ourselves onto the cloud platform. So now everyone, we know about the some clouds, right? Simple, the cloud we know about, right? Everyone, whatever the we are getting AWS, Google Cloud, we are getting Microsoft. So there are the different resources they are providing the cloud environment where you can develop your pipeline, where you can work, where you can take the input, where you can actually process, and you will provide the output. So for that efficiency, reproducibility, these are the different resources which we are getting. But now, as an bioinformaticians, as an biological researchers, we have to more focus on the data which is belong from the our resources. So mostly we know about NCBI, which we are getting the data, EMBL, Uniprot. So these are the open access resource where you can download the data, biological data, and you can work on the cloud platform. So for example, the platforms like the Google Colab, we can say AWS, TeraBio, right? These are they are allow the run analysis without needing any local machines, right? Our systems we call the local machines, but we can run some pipelines on this. Yes, you will get some free access also. You can just try to work on that. Definitely you can work on the different services and try to implement as a students also. So these are the, the cloud platforms. The DNA nexus, it give the information about the biological data handling. Yeah, as I stated, NCBI, European Nucleotide Archive, and Gene Expression Omnibus. So these are the having the rich repositories to produce, to give output in the omics data sets. You can just visit and you can download the data and take these data for your understanding. Next, as I stated, the AWS, GCP, Azure. So these are the different resources they are providing the cloud bioinformatics services. So yes, you can create some academic license for 1 month, 2 months. You can just get some tokens on that. Just work on that. You can try to understand as a student also. Along with that, you can work on the open target database and Uniprot. So there are the different types of the initiative where the bioinformatics forums has been taken to identification, the functional and the disease annotation tools also. Yeah. So these platforms, they are specifically helpful for the students, researchers who want to explore without any heavy infrastructures. Yeah, next we need to understand. See, we need to understand about what are the different types of the bioinformatics tools are needed to learn. Yeah, these are the free resources where you can implement these tools in your projects work, in your internships, or your understanding to make the industrial based skillings. Yes, these tools are also applied in the industries. So yeah, already I think the data pipelines like that has been discussed how to like we can start our journey from accessing the fast queue data to conversion of VCF, and later from the VCF to the downstream analysis. We have the identification of different variations, clinical variant annotations, prioritizations, right? There are the different types of the downstream analysis you can see. Along with that, you can see the visualization using IGV, and also you can implement the R and Python, the different libraries to visualize these data. So if you want, like you can start a journey, you can just take a simple, yeah, but all these things I recommend everyone try to work on the more on the Linux environment, because these tools are very important and very easily installed on the Linux environment. And as an bioinformatician, if you want to start your journey, work more on the Linux environment systems. Yeah, category, if you see the quality control, the fast QC, it's a very important tool which has to be use case accessing the raw data. Before, this is the first steps where you are accessing the data from the sequencer, okay, and you have to read the quality. After that, you can go for the next steps, that is the assembly, alignment, and conversion, and the different downstream analysis. So this is what the first tools we have to know about the fast QC. So in that, you will get to understand what is the Freight scores, how this k duplications, if the part of the sequence is completely different. So you have to understand first these tools. If you're working on the alignment, you can work on the histart and the star, that is the RNA-seq data analysis. Yes, workflow management. So now nowadays, everyone is working on the workflow management. That is the Galaxy. Galaxy is open source tool where you can work on these. You will get, uploading the FASTQ file to a generation of the VCFs. You can define some workflow like first step, second step, third step, like this. So this is what workflow management. Along with that, you can use the different Dockers, you can use the Nextflow and the SnakeMake. These are the different workflow management systems are available. But Galaxy you can try as a beginner because it is a web-based platform to reproduce your pipeline. Data analysis, yes, obviously data analysis, because whatever we are getting the data, it has to be analyzed in a biological meaningful manner. For example, we need to calculate some analysis scores, risk scores, patterns, that is the statistical computing, scripting we require, and data manipulations. And if you are working on the RNA-seq, that is the single cell RNA-seq analysis and its visualization, we have to more focus on the Seurat. And when you are working on the biomolecular interactions, so basically cytoscape, it is a tool which is used for the network analysis. So this is the snap of the FastQC, because if you once you install and once you understand about how to work on this FastQC, you have to take the input from the FastQ file. Later, this is a FastQC file. It is a widely tool for accessing the quality of the raw sequencing data, which is coming from the high sequencing platform. For example, Illumina. Okay, it provides a complete visualized summary, which includes the read quality, GC content, along with it get the adapter contamination and the duplications level. So this initial quality, it helps to research decide whether the data is suitable for the downstream analysis or it need to be pre-processed, like the trimming and the filtering. So once you're done, you can go for the next level of steps, which is has to be performed in the data pipeline. Next, I'm not going to focus on the complete RNA-seq data analysis, but here, if you see the fast speed and best accuracy, and you will get, alignment based and alignment free. So you have to process trimming, alignment, quantification, and differential expression analysis. So in that, like HiSat2, we call, and the SHARP. So basically, these are the aligners which are used for mapping the RNA sequence rates with the reference genome. So HiSat2, it's known for the speed and its memory efficiency, so which make to align with the large data sets. While Star XL, it's handling the splices alignment, and it is also preferred for the transcriptomic studies. So both these tools, they are foundation when you are working on an RNA-seq pipeline, and also it enable accurate quantification of the gene expression. Yes, next is the Galaxy. As I stated, it is a web-based platform where it allows user to be perform a very complex bioinformatic analysis through the graphical user interface. So it's if you are working more fascinated about the graphical interface, please try this without needing to write the code. Yes, also it supports a wide range of tools and the workflow which is already designed in the genomics, transcriptomic, and metagenomics. You can utilize those workflow for your data analysis. So Galaxy is very promoted for its reproducibility, and also it is collaborating by enabling the users to share the workflow and data sets very easily. Cytoscapes, this is used for majorly, we call it is a powerful platforms when you are visualizing the molecular interactions and its a network. Okay, so biological pathway we know everyone. So it allows to integration of the various data types, which include the gene expression, protein-protein interaction, and network modeling also. So researcher, you will be use these cytoscapes for explore more the functional relationship and also identify the key regulators and perform its enrichment analysis, specifically when you are working in the system biology and the pathway studies. Yes, R and Python. Now we know about everyone. You are working on the data analysis, data science, right? Each and every field, AI and ML, this technology has been emerged, right? So to understand AI ML, Yes, as in biological, if we don't know about the background of any programming, you can start your journey with the Python. Okay, so try to understand first how it is working, understand basic concepts, what are the basics, syntax, it's a basic data structures, how to handle file. Once you understand, shift yourself into the R. It is also similar. Okay, we just need to take the difference. What is the difference between the R and Python? What are the command based difference? Yes, both the programming, it has the lot of applications when you are working on the data analysis in the biological bioinformatics, because it provides the different types of the modules, it provides the different types of the packages for working the statistical analysis and the data visualization. For example, if you see the bioconductor, ggplot, and there are the different libraries. And also in the Python, if you know, it is very popular for the versatility, and it's very easy for making the automations, data pipeline, integration with the machine learning and data processing. Okay, which we have the pandas, numpy, scikit, matplotlib. So there are the different libraries which help. So both these programming, they are help to write the code, to write the scripts, making its automations for the custom analysis. So yes, in that, in R, we have the Seurat package, which is used for understand the differential expression analysis. Basically, the R has to be having, more powerful to identification of the cell type of the identifications, trajectory inferences. Also, it is making to a very, working on the single cell genomics and transcriptomic analysis. Now, here, how to start to work on the learning the pipeline and scripting for the omics data. So we understood here what is the omics data, what is the, the clouds which we can use here. So yeah, as we already discussed, we need to start on the Linux, write some script, that is the bash scripts. This particularly bash scripts, it is working on the command line basis. Start to first working on the Galaxy, later you have to shift your on the Linux, start to basic commands and try to work on the scripting. So once you're done with the bash scripting, definitely you can move to the next, that is SnkeMake and the next flow. If you see nowadays, these are the emerge, these are the trend in the bioinformatics to making workflow management. So here, what it will use, it is integrating the multi-omics data, make the genomics, transcriptomic, and proteomics. So you have to utilize the SRA database, Geo database to understand, to take the the sequences for your pipeline and make it integration with the SnkeMake and the Nextflow. We have to practice rebuilding the pipeline from the public GitHub repositories. So whatever the Git repositories they are getting, just try to rebuilding the pipeline. Also, you can use the JJupyter R Markdown for making a literate on the programming. So as suggested, how to start with the learning, these are the steps. Also, along with that, you have to more focus on the annotations, wherein filtering, data visualization, and downstream analysis. So these are the steps which you have to follow. Along with that, create the Git repositories, create the this understand the programming notebooks. So also you can take a participation on the BioStar, Stack Overflow. There are the different bioinformatics community which is very supportive for to understanding this pipeline. Now, next, I'll back here on the open source and commercial tools. Yes, there are the both the approaches having its own advantages and disadvantages. So, as a students, we have to more focus on the open source. See, the cost, if you see, open source is very free, right? But commercial, we have to go for the some subscription models and the paid. In the open source, it has this strong the portfolio which we are getting, the GitHub, we have the forums to join. But when you are working on the commercial tools, we have to take the vendor supports. The customizability of open source has high, but commercial is a variable. Ease of use, when you are working on the open source, it may require some coding, but commercial tools, it's require, it is more user friendly. So the entrance, they can should start with the open tools, the Galaxy, R, Python, and Seurat. Okay, so these are the free, flexible, and widely supported for the community also. The commercial tools are very ideal for enterprise and if you're working on the regulatory environment. So there are the liquid teaser, pathway analysis, gene spring, which offer more polished inferences. And also this supports, but have the some cost. For students and interns, I recommend starting with the open source tools, which are very powerful and they have help you to build the foundational skills. So yes, this is the what you have to work on the active user communities and the documentations. So final tips for the students and interns. Yeah, it is has to be joined with the different bioinformatics forums, join with the biostars, Stack Overflow, and the different types of this library. Also, you can contribute the different open source projects to sharpen your coding skills. Also, pick the participation in the different hackathon coding challenges. Try to work on this. Follow the GitHub, which is trending in the bioinformatic repositories. Also, you can take the different online internships, which is provided by the different resources. You have to embarrass to make the reproducibility. You have to more on the version control. Here, I'll suggest you can learn the basic starts from this Python and R, practice the data visualizations which help to communication of research, understand the biological context which is behind this data, and try to work on the small projects where you can get hands-on experience. And also you are getting the EMBL, there are the different certification to understand the basic bioinformatics, which has to be added to your resume also. That is what you can start with the internship, practice, learning, experience, and you will get the success. So, yes, thank you for your attention.
Thank you so much, sir. That was incredibly informative and practical session. Thank you so much once again.
Thank you.
So, actually, sir, I must say that your deep insights into the essential tools, platforms, the scripting strategies for NGS, and the data analysis have truly added immense value to this webinar. And I am truly thankful to you for joining us. I'm
Thank you.
sure our attendees now have much clearer picture on where to focus their learning, especially with the fast changing demands of 2025 in mind.
Yes.
So, yes, thank you, sir. And I would request you please stay with us, and as we proceed for the third session, and after that we will continue with our question answer. And definitely, there are many students who are already posting questions. So, attendees, I would like to tell you that we will take up your questions. Maximum number of questions we'll pick up, but that will come after the third session. Okay. So please stay with us. And now let's proceed into our third and very exciting segment where we will be diving into internship projects. I can already see some students have asked questions about internship projects, where to start, and which are the companies that are offering what kind of projects to work on. So, guys, we have that for you. Okay. So, it's my pleasure to introduce our next speaker, Dr. Ilamati Natarajan, a highly accomplished bioinformatician and an educator with a remarkable track record in computational biology, data analysis, and genomics. Welcome, ma'am. Good evening.
Good evening, ma'am. Good evening, everyone. Yeah.
Yes. So before, ma'am, I hand over the controls to you, I would definitely like to introduce you to our attendees. Uh, so, as I told you guys, Dr. Ilamati, she holds a doctorate in bioinformatics from Dr. APJ Abdul Kalam Technical University, as well as an MBA in Information System Management, a combination that reflects both her scientific rigor and strategic vision. She has served as assistant professor and HOD in charge or at Kalinga University, where she led academic and research initiatives and with excellence. Her technical expertise spans bioinformatics, pipeline development, quality control, machine learning applications in genomics, and much more. She is also recipient of Senior Research Fellowship from Indian Council of Medical Research, that is ICMR. Definitely a recognition of our impactful contribution to the field. And today she will be sharing how to select meaningful internship projects, cross disciplinary project ideas, and how to present your work effectively, including tips on documentation, GitHub portfolios, as well as communicating your research with impact. Okay, so please join me in welcoming Dr. Ilamadi Natarajan to take us through this final but extremely important segment of our session. Over to you, ma'am. I'll pass the control. You can start out your session.
Yeah. Thank you so much, ma'am. Yeah. My slides are visible.
Yes, ma'am. There. Please continue.
Yeah. Okay. First of all, I would like to thank Titi ma'am for such introduction, and also I would like to thank my other speakers like Ms. Tigda and also Dr. Yogesh on throwing such a spotlight and in-depth analysis about the NGS in very detail about the tools and techniques in much detail. And I will like to especially thank the audience who joined here today for to know more about the NGS session. To all the attendees, I would like to thank them especially for that, for joining this useful session. So here, right now I'm, in front of you, I'm like a scientist. But once upon a time, actually, I am a, job seeker like you. Okay, where I was looking for a job where I'm one among of all of you. Okay, so after my graduation, confused graduate, and after my doctorate, I think what next to do. Okay, such kind of position even I am there. So I put myself my own experience, and also luckily, I got a chance to like guide many students and also some working professionals in the field of bioinformatics, computation biology, and AML, especially in guiding the internship. So I would take their experience also, how they handle the internship, what to do in the internship, what not to do in the internship. Okay, so that things I'll share with you today. And also nowadays we are in a race, okay, for getting a job. Okay, so huge peoples are behind in the race, and much more competitive world we are standing. Okay, so everybody has CV, everybody has skills. Okay, but how to we make ourselves stand out from them. Okay, so I'm more excited to tell about my experience and also the my colleagues or my own students experience over here to make it much more meaningful internship, how to do. Okay, so that we are going to see this exciting segment today. Okay, so generally internship is it is your research identity. Okay, so you should develop it very well from the infancy stage. Okay, and you should make it remarkable. Okay, so how it can be done, definitely I will tell you, and share the interesting experience. It won't be a boring lecture, it will be a interesting experiences from all of us. I'll be sharing this with you. Let's move on to the thing. Okay, so first of all, why does this internship matters? As I said, nowadays every people's like they are trying to do the interdisciplinary course. Okay, and they try to put the skills in the CV. Like I am skilled in Python, I'm skilled in NGS data analysis. Okay, so everything we are making. First of all, one important thing I will tell is that you should put what actually you know. Okay, so we should be very honest with the stakeholders. We should tell what really we know, what technical skill we really matters. Okay, but you can ask me like by using this honesty, do we get the job? No. Okay, so you should instead of you speak in the interview, your CV should speak actually. Okay, you should not scream in the interview that I know this, I know that. Your CV should speak. Okay, as I said, putting filling up tools, all the necessary things or necessary keywords, that doesn't never going to attain a job for us. Why? Because you need to show something in action. Okay, so that will matters a lot. Okay, so hands-on experience is one important thing which really you need to have it. Okay, how it can be proved? Okay, even at early stage of your career, if you show internship is not just like a any project. Okay, it is your early stage of your experience. It matters a lot. Okay, so your experience through this internship, you should do it very carefully. And after all, whatever you do, you should document it very carefully. Okay, you can publish a good papers out of your internship, and also you can store your whatever the pipelines you have developed. You should definitely prove to the world what you have done. Okay, so you should make a repository where you should store it in the GitHub. Okay, and also necessary collab notebook, everything you need to be put over there. Okay, so now instead of your publication will speak, your internship will, what actually you have done. You can, okay, knowing the tools, how practically you have implemented with solving one particular problem, that matters a lot. Okay, so this will take you to the standing out from the other people's. Actionably, your CV will speak over there. Okay, so the first step of your early career is internships. This matters a lot, whether it is you're doing an MSc project or BSc project, whatever you do, that is a starting point of your research infancy. Okay, so that should be very good. Okay, so here internships will tell you that how easily you can handle, even in a 3-month internship, 1-month internship, 6-month internship, 1-year internship, whatever you're doing, that is your experience. You can show that as an experience in your career. Okay, so you no need to stand blank and say that I am just a fresh graduate. Okay, you can say that I have done a hands-on internship. I am proudly, you can say that I'm an intern. I have practically implemented my skills, knowledge into skills I have turned up. Okay, that you can actually show. Next thing is that you can definitely take a more important tools you can take. But whatever the tools you take, please think it whether it is necessary for my internship. Then you took that kind of tools and relate it to your internship very effectively. You please do that. Okay, so then say every people, recruiters or any, say if you're going for academic role, okay, every kind of academician looks that whether a candidate is suitable in handling the project. Okay, whether they done a good real-time data they have done or not, that they will check. Even the industry recruiter or any kind of recruiter, really they think of, if I'm investing in this person, did he or she can able to handle the real-time data? That's what they really think of. Okay, so on this basis, I would suggest that this internship will build you the confidence, and also it will throw you a right direction, and also a good collaboration and networking you will get when you are performing the internship. Yeah, so how do you make your internship project very impactful is that there are certain core elements you have. Okay, first of all, you should understand what is your biological question you are speaking about. Okay, so when you see computational is one thing, now it is very unavoidable. Okay, in life science, computational biology, bioinformatics, AML, all these things are now, it is unavoidable one. Okay, so you should be, for data science, you should be very expertised in these skills in the life science to analyze your and interpret your results in a very effective way. Okay, so first of all, you should understand what is the question I need to do the research. Okay, you should define your biological question very effectively. You should frame your research hypothesis. Okay, so next thing is that nowadays, generating a data set and our publicly available database, which give us a advent to opportunity for every kind of researchers worldwide to take those real data sets in a free in free format, like no need to pay anything. You can freely access the real-time data set for your effective project. You can really take and do the analysis on your own. Okay, so we know, like in the previous lectures you have seen, developing a good pipeline with the integrated tools that makes you and good popular tools you need to use in your work. Okay, and it's not about developing a pipeline, whatever the result it is generated with the pipeline nowadays. Okay, so you need to interpret really with your biological question. Is it matching? You need to look back. Okay, it is not about just generating a code or anything. You need to answer the biological question, whether your objectives are fulfilled or not. That need to be effectively communicated. Okay, so whatever pipeline you develop, you should document properly. Okay, either you can write a blog on that, you can put a LinkedIn post, or else proper official way of doing it is GitHub or Google Drive. You need to store them. Okay, all your thing. See, not only pipeline, along with the detailed description of your work, you can publish over there. So with that GitHub, that link, you can publish your scientific articles. And also there are many preprint servers are there, like BioRx we have, so where you can put your impactful research over there. Okay, yeah. So if you see, what is the actual project I should do? This is the question which comes to everyone, not only the students, also experienced professional, when they want to transition their field from one field to another field. Everyone has this question in this. Okay, so nowadays everybody know NGS is a trend. Okay, and also the multiomics project is a trend, we know. But in that, what is the project to choose? Okay, so you see what is more relevant, okay, in the clinical aspect, and also with the working with the real-time data set, which your project should reflect bioinformatics perspective, coding perspective, and also the pipeline perspective. And with it should answer the, say, whatever research we do, ultimately it should directly or indirectly, it should prove some value to the society. It should add. Okay, so in that basis, I feel that we should choose a right project which can help the society also. Okay, so most few examples I would like to throw here, but many more such good projects are there. Okay, few examples like breast cancer data with RNA-seq, okay, you can take, or else like analyzing the gut microbiome in obesity, okay, we can take, or else like performing in neurodegenerative disorders like multiomics data or single cell RNA sequence data. Such examples can be done. Okay, but much more examples are there. Okay, so when I could see, as I have told, I have guided many students also, and also many peoples I have seen. Okay, what is the common like mistakes they generally do in the internship is, they are not clear with the biological objective what they are need to work on. Okay, so peoples are like, I can say that they are like a, uh, they want to work with more tools. Okay, so the students want to work with more tools. Okay, unnecessarily they will add the tools in the internship which is really not required at that moment. Okay, so you should avoid that mistake. Okay, and I have seen some students where they get the pipeline, they just copy paste the code without understanding any logic. Okay, that things really you need to avoid it. Okay, you should see, initially as a beginner you doesn't know about the code, you can take the pipeline, you can copy paste them, use them, but later on you need to understand why I'm using this command, why I'm using this function. You should understand that particular portfolio, you need to understand. Okay, and next thing is, whatever you do, even a piece of work, small piece of work, you please document it. Don't you don't ignore the documentation. Okay, and also in GitHub, which version you are establishing first, and next which version you are establishing, such version control you should have. Okay, and also you should develop a reproducible pipeline. Sometimes pipeline works, it good in, especially in AML models, I could see students develop with 99% accuracy. Afterwards sometimes 75% accuracy it says. Okay, but such kind of things should not be there. You should be very consistent, a reproducible pipeline you should develop. Okay, and one more thing I would like to tell as an example. Nowadays people are becoming like a pipeline zombie, so adding so many tools which is not really needed. Okay, so see, zombie means what they do, they eat more things which is not really required, right? So whatever we could able to chew, we should take that kind of food, balanced diet, right? Same way here in internship also, you need to add the right ingredients. You need to add which tool I need to use for my objective, only that you need. See, learning is one continuous process. Lifelong, we should learn about, as a life science researcher, we should learn more about the tools and also softwares, everything we should learn. But as here, if you see, in this particular thing, in internship, you just add only which is needed for that. Avoid which is unnecessarily, which is not needed. Please don't take too many data set and tools. Okay, but whatever you make that, even a thing, whatever you have made, the internship should be very impactful. You should make that. Okay, and some students, they do decision on their own, but please, you be in consistent communication with your mentor, and you please be on loop. Okay, with them, whatever step you do, inform them, because as an experienced person, they can guide you in a better way. And next one I wanted to tell, okay, how to choose the right project. Okay, I will tell you few tips for the same. Okay, so first of all, you identify what is your target interest you have. Okay, whether it is cancer biology, agriculture field, animal field, vector biology, okay, or AML, as a developer you want to do, tool developing you want to do. Especially you identify your main interest and sub interest. Okay, based on that, next thing you need to look for is the data set available or not. Okay, publicly available, or it is cost communicating, cost efficient. You need to check all these things. Okay, next thing is, you need to split your work. Okay, you can split your work, like correct timeline duration you need to give. overnight you cannot do a project. Okay, and don't do a one simple project to a lengthy time. You give a manageable time, like for one task you can apply 1 week. Okay, so or 2 to 3 weeks. Sometime you can require some project to be done in 3 months, 6 months. So for every task you make a clear strategy of phases you need to make. Okay, then you need to see in your project, add what all the tools already related to the project, and you can also welcome to add some new tools which is really required that you can add. Okay, so then next one more thing, initially as a beginner we may not aware of all kind of work or anything. Okay, so you can take the pipeline, you can use that, but you can do minor changes in that according to your work. Okay, you can practice, and then later on you can establish your own well developed pipeline you can establish later. Okay, next one important aspect which everybody need to think of is choosing the right mentor. Okay, so how do you choose your good mentor? See, your mentor should be definitely skilled enough, subject expertise, okay, domain knowledge, and also the tool skill they should have. Okay, and they should also having experience in guiding the intern and also the undergraduates, students, freshers, and working professional. Every much experience they should have. Okay, and next one important factor, they should be available, and frequency of availability and giving the feedback, that should be on right time. They should be there with you. Okay, then you can also look the expertise of publication, how they did. Okay, and which research category they have did. You can evaluate all those skills also to choose the right mentor. Okay, yeah, so where do you find the mentors? Basically, if you see, nowadays we have lot of research institutes in India and abroad, as well as you have academic institutes in India and abroad. We have, okay, nowadays we have a professional network, LinkedIn you can choose, as well as you can also develop in GitHub. You can choose such kind of good peoples, and you can connect with them. But it is not and definitely lot of institutes are providing nowadays the choice of internship. Likewise in biotechnico also we provide lot of courses with good internships we provide. Okay, you definitely look for the course where the internship support is also provided. You can through that, and you can choose such kind of institute for your course and internship section. You can choose that. Yeah, so next one more thing which I would like to say, nowadays people's recruiters, they really look for one person who is a whole jack of all trade. Okay, so you should be master in all. Such kind of person is required. Okay, you should be a single master, can handle everything. So you should be a life science person. You should be good in programming. You can able to solve that thing using the computational skills and also your AML skills. Such kind of person every recruiter is looking for. Okay, so you should have in internship, you should prove that. Okay, interdisciplinary projects you should do, cross disciplinary project ideas you should throw on with your interest. You should throw on that. Okay, for suppose if you want to work in a cancer genomics, okay, you can always integrate AML field into that. Like suppose if you want to work on plant transcriptomics, okay, you should look for a hard trend of the topic. Okay, and then you can apply the computational field along with the coding, everything you can apply over there. Okay, for suppose if you want to work with microbiome, okay, so there you can also integrate metabolomics field over there. Okay, now when it comes to the multi-omics, all techniques, omics technology can be integrated together and perform a very good analysis can be done with your projects. Likewise, like epigenomics, here also you can have a blend of machine learning and coding can be applied here as well. So nowadays your project should looks like bioinformatics, AML, coding, and with the right solution, right tools, and right data set with a real-time data set you need to include in your internship. Okay, yeah, so we have seen that lot of real-time data sets are nowadays available in all these databases, which is really free. And also we have you can use any of the user friendly graphical user interface tools, or else you can develop your own pipeline integrating all of them together in your codes. Okay, that is also possible. Okay, so everyone, please just don't forget, whatever you do, even a piece of work in your research, please document it. Okay, and one more proverb I would like to say, as a researcher you should publish or perish. Okay, so whatever you do, piece of work, try to publish it. Okay, otherwise you need to be perished. Okay, so keeping in that in mind, so you make your GitHub portfolio very stronger, so that you should see your GitHub portfolio in your CV, that should speak, and your internship will add very much it will give a special portfolio to your CV. Okay, so CV should speak there. Okay, in interviews. So there are few elements very much really needed in the GitHub page, that is like readme, Okay, so readme files where summary of your project, tools, goals, outcomes should be there. And also you should have add all the results, visuals, tables, final report, everything should be added. And you should also have a notebooks which is very cleaned, okay, commented one. It can be either Jupyter notebook or R Markdown, where description along with your codes, everything can be there. Okay, and also you should add your raw data and process data and necessary links. What is the suitable link you have chosen? All those things necessary. If data is large, you need to upload all the details. It should be very transparent actually. Okay, yeah. So how, see, you have done a project. Okay, next thing is how to make it, your project should be visible to everyone. How you can make, first of all, if you're doing dissertation, definitely you will do the internal presentation in your lab or college, you will make it. Okay, likewise, you can also make some good posters or abstract in the conferences, you can make it. Okay, like in many of the good communities, international conferences, Indian related good conferences, you can present it. Okay, and also we have online tools where preprint servers are available. As I said, before publishing it, you can put your papers over there. Okay, in bioRxiv, you will get the ID, proper ISSN number, everything you will get over there. Okay, and main thing is you can store it in the GitHub repository. All your pipelines need to be stored over there. Okay, so where other people can note your work, okay, they can do your work, cite your work. Okay, that also matters a lot. Okay, and next is finally, as a report, how to present your project tools, that we need to see. Okay, so here we have a biorender. Okay, where all kind of results in a good scientific communication, how it is required, good project report kind of thing. Finally, it could be, we also have Canva, where infographics along with the images, all information can be uploaded over there also. Okay, next we also have a Zoom link. Okay, where you can give a 5 minutes demo about your profile, and you can video profile you can make about yourself, and you can put it in your explain about your internship. You can make that video profile anywhere to share while sharing the resume. You can share that also. Okay, so you should see even the complex project with clarity when you explain. Okay, so you should always present it with a good story, interesting twist. Such kind of arcs should be there in your project. Okay, as a story narration, you need to tell about your project anywhere. Yeah, so as a final takeaway, I would like to tell, please remember. Okay, so first you think of what is the real question of your research. Don't think about the tool. Later on, tool learning is a continuous process. You can learn anytime. Okay, so you just pick a project which really fit your interest. Okay, and also is the time duration, you give the enough time duration for that, and also you improve your skill level according to that. Okay, choose a right mentor. Okay, and also you choose a right data set and right real time data to work on. Okay, and also you can catch some cross disciplinary hot trend ideas. Okay, what it is going on in industry. Okay, or scientists what it is looking for, recruiters what it is looking for. Please of their attention, you can choose some project related to that. Okay, so whatever you do, finally you upload your project in GitHub. Okay, you make it happen. Okay, yeah, so internship is one thing which really show your personal research showcase over there. Okay, so make it to speak your story for the success. Okay, thank you so much patiently for this session. Any questions? Everybody can please go ahead.
Thank you so much, Dr. Ilamati ma'am, for that truly inspiring and insightful session. Thank you, ma'am. Definitely your practical advice on choosing the right kind of internship projects, and more importantly how to document and present them effectively, was incredibly valuable. I'm sure very many attendees are walking away with a clearer sense of direction and confidence in how to build a project that not only adds value to their learning but also strengthens their career profile. And also I must say that your warmth as a mentor combined with your depth of experience really came through today, and I know our audience is grateful for the clarity and motivation you have provided. Thank you so much, ma'am.
Thank you, ma'am. Thank you, everyone. Yeah.
And I must thank all of our amazing speakers for those wonderful sessions. And for everyone who are still with us, just a quick reminder, remember our exclusive freebie, which is packed with internship resources and learning links, is coming up right after the QA session. Okay, so stay tuned. We are almost there. Now we are going to open the floor to take your questions. I already have some questions with me. And also I request everybody, you must have noted down your question, so please you can go ahead and ask your question in the chat box and we will pick it up. Okay, so as just now our team had displayed the our internship project or the course which is going to start from August 4th. Yes, Ningthao, yes, your question. Is the NGS internship project will begin from August 4th? Yes, this project is going to start from August 4th. Moving on. Okay, so another question that I have here is from Vice Chapter. What the language should I learn for this? I think he's asking about what kind of language is needed for NGS data analysis. Stingda ma'am, can you please give some clear direction to this attendee?
Hi, Wise. What I'd like to say about this is go ahead with one language. Master one language, which is very important. Whoever masters one language, it is seamless to understand any pipeline, any imports, and any visualizations in any other language. So first, if you're a beginner, please, my suggestion would be master any one language, either it's Python or in R, or any of the language that you are comfortable in. When you are comfortable, and once you master that language, then you can seamlessly go ahead with this test, like stats analysis in R, because R gives you more plugins with statistical analysis. It will be very seamless for you working with bash profiles. The basic Linux also, is very important to understand. So basic Linux and any one language that you master, and then it is very seamless to understand any of the plugins and the one version controls and any of the imports that is coming in. So I hope that answers your question.
Thank you, ma'am. And next question we have from PW Need Motivation. Sir, please suggest some good government internship opportunities in 2025 that can build my career. Yogesh sir, can you please help our attendee here?
Yes, Puneet, motivation. There are the different types of the government internship programs are there, but I just want to understand what's your bachelor or master. Because if you are aware about master, if you have done your master in biotechnology or bioinformatics, there are the different government sponsored programs are there. That is the BITP, BIITP also is there. And along with that, if you are working on the Tata Institutes, you are getting the different types of the fellowships as and working as a research trainee. So it depends on your like what your BTech, MTech, or like what's your bachelor or master. Based on that, you will get these different types of the internships opportunity.
Yes, so guys, you just have to do a little bit of more research, okay?
Yeah.
And yeah, Dr. Ilamati, do you have something to add to this, ma'am?
Yes, ma'am. Definitely, I will tell. Like lot of openings will keep coming actually. Okay, so internship opportunity they keep come on LinkedIn, and also the even our biotechnical site we keep telling about that, and also other kind of career site they can keep noting on that. Every year, 2 times the call will come. Okay, so based on that, they need to apply for such kind of internship. Yeah.
Yes.
Thank you, ma'am. Thank you, sir. Next question I have from Abdul Mubarak. Now, this is, I will say this is a question which everybody is asking. Okay, it's related to AI. So his question is, my doubt as bioinformatician, will a bioinformatician be replaced by AI in future or not? So, ma'am, I would like to know, ma'am, sir, I would like to know your point of view, like from everybody. So, starting with you, sir, can you please put some light on this? What
Yes,
are your thoughts?
actually, yeah, yeah. So yes, Abdul, it's a very good question because now everyone we are in the era of AI and ML. When we are started our journey, that is understanding some single cells and single gene and single protein. Now it's completely drastically changed. But if you are as an bioinformaticians, everybody now we are working on how the AI and ML it is implemented in the biological data sets. So it not we can say it is not replacing because if you see there are the manual or human experts is also required for to understand this data and to working on this analysis of data. my suggestion for you to understand and upskill yourself to try to work on more on AI and ML technology, how to be integrate in the biological data sets. So yes, there is no any, we can't say it's like not replaceable. We have to upskill ourselves according to the trend and according to the technology.
Thank you, sir. Snigdha ma'am, what are your thoughts on this?
Ma'am, I 100% I'll go with sir's what sir has said, that you need to upskill. You need to always upskill your domain, your expertise, your skills. AI cannot replace the human critical thinking and the analysis. The only thing that we can as a bioinformatician do is upskill and move along with what big data is coming, ML and AI is coming. Use it as your tool, your analysis. Just use it under your expertise and critical thinking as humans. So definitely what sir said, I'm 100% I'll go with him, right?
Thank you, ma'am. Dr. Ilamati, what are your thoughts on this?
Yeah, it's a very good question. But I would like to say right now, it is an era of bioinformatician, I would say, because from wet lab, a lot of large data sets are there. So now we are bioinformatician is a master to play with the AML. Okay, so bioinformatician can understand the biological problem. Say if you go with a computer AML engineer, he can develop something. Okay, whatever we tell, he develop a code or tool. But ultimately, they need to come to the bioinformatician to develop a tool which can solve the biological issues. So we are the masters. Bioinformatician is a master who will handle all kind of AML or thing which need to be rightly skilled, okay, with the life science, and also balancedly we need to do that. So we will be the totally master. Bioinformatician cannot be replaced, but it is a right era for us in future also. Yeah, I would say.
Thank you. Thank you so much, ma'am. So, yes, guys, so that's a very valid question. But yes, the same thing applies for, I think, any kind of skill that you have to change. You have to reskill yourself. You have to learn the advanced things which are on market. So, right now, it's the era of AI. So, AI is not going anywhere, and AI is there to make our work even more easier, I will say. So, we have to learn how to use it the best way we can. Right. So, you have to understand, you have to learn those skills, you have to reskill yourself in that way. Moving on, so I have next question from Paras. So he's asking about how to save our data in GitHub, and how can I save it in public everywhere, and or should I make it private? So actually, Ilamati ma'am has already given you an idea about how to go ahead and do the things in GitHub, but I will still ask Snigdha ma'am, can you just help him a bit in this particular
um,
technicality?
So hi Paris, as Ilamati ma'am has already described, how to commit and how to save your files. Moreover, I'll encourage you to, it's your choice to be, to uh, keep it as as a private thing, or as a public. Being sharing it, or on a public platform. Of course, people can cite that, read that. You can just collaborate your work with with scientific community and everyone. So it's on you that how you keep it. But moreover, what ma'am already explained, that use descriptive commit messages for your for your GitHub repository, that whenever you go back, it is easier for you. Tag versions as well, because working with pipelines, it is very important to work with the version controls. Your readme .md file should be updated, whatever overview, whatever installations you had, because whenever the sample size or the data you want to extract, and how you did that, is always saved. So all of that will help you saving your GitHub repository. And making it as a private or public is your choice. Making it public will definitely cite your work. Sometimes people can read that, share that. So go ahead of whatever you want to represent your work as. So I hope it answers your question.
Thank you, ma'am. Yes, Paras, I am sure you have now understood how to go ahead with GitHub. Next question we have from Dhruv. He's asking, give a roadmap on bioinformatics, please. So I would definitely request uh, our technical team. Can you please display this question? Give a question from Dhruv Sharma. Give a roadmap on bioinformatics. Yes. So Yogesh sir, can you please give him a guidance on how to create a? I think he is asking about career roadmap, how it should look, and how he has to plan towards a successful bioinformatic career.
Yeah, hi Dhruv. It's a good question. So basically, if you are in the bachelor of the any life sciences, you can start your journey in masters to understand the bioinformaticians. You can go for the masters, or even though if you are in master in any field, if you are for the life sciences, start to working on, first understand what exactly the bioinformatics, what is the trend of bioinformatics, and start to working understanding first biology, because if you are understand the biological concepts, because bioinformatics it solve the biological problem. So after once you're done, you can start to working basic databases, sequencing tools. That is the basic bioinformatics, and again you will go for the advanced bioinformatics. After that advanced bioinformatics, you will get the different domains. For example, you will get the computer drug designing, you will get the genomics, transcriptomics, and different the domains you will get according to your interest. Later you can implement any scripting language, start to working on that scripting, and once you're done with that scripting, you can implement those scripting knowledge in the bioinformatics to solve the biological problem. So this is how you can start to, the roadmap of bioinformatics. I think, ma'am, she will give the detailed understanding about the internships and all this thing, what the Biotechnica is providing. So I can suggest to you, okay, if you are belong any stream, any life sciences background, try to connect with the team. I think definitely they will counseling about the different types of the offerings about the internships, so you can start to understand from the basics of bioinformatics, and definitely you will get, a knowledge in the bioinformatics.
Thank you so much, sir. Thank you so much for helping Dhruv, and I'm sure many students also, many attendees here also have a similar kind of question. Thank you so much for answering them. Next question we have from PK, and it is a very general question, I must say. So he's asking that I don't know any language like R or Python. So how is there so I think he's asking like, will there be any problem while learning the NGS data analysis course if there is no background knowledge of R or Python? Dr. Ilamati, you have worked to design the curriculum for this NGS Multi-Omics internship. So I would definitely ask you, ma'am, do you think will that be a blockage to understand the process of data analysis?
No, ma'am, not at all. Like, I would like to say, like, see, you doesn't know about that. That's why you have chosen the course of internship. So definitely, it never hinder any places. Okay. So here, not only the coding you will be learning, but right use of Linux, and also what is the right tool and softwares. Okay. So you will be learning here. Okay. And one more thing, during the curriculum, you'll understand the basic. Okay. Lot of demonstrations also will show specifically. So there also scope to learn, and also when you do the internship on your own, you will learn more also. So here nowhere it will it will hinder your thing in the course. Only thing is you should have a special interest, okay, in practicing the codes, okay, since you need to practice more with the tools and softwares, any of the bioinformatics thing which is related to NGS. When we tell, you should have the only the self motivation, and also we'll also tell the right thing to plan actually. Yeah. So yeah, so you can definitely, it never hinder your course actually. Yeah.
Yes, thank you, ma'am. And I will request our technical team to please display the our course that we are going to start on from 4th of August, next generation sequencing and multi-omics data analysis internship with project work. So we will be providing 30 days of training, and I think Maria, you had asked whether we will be giving you a detailed course on R and Python. So no, we'll not go in the depth of R and Python, because for that we have other courses for that particular thing if you want to learn R and Python in depth. But yes, how it can be used, how those tools can be used for NGS data analysis, those kind of trainings will be given to you with proper demo sessions. Okay. And our course also includes 2 months of project work where you can work, and you can also you will get your hands-on experience of trying out and practicing those tools, and that definitely adds up to your resume. Okay. So with this, I think it's time for us to close today's session, and I would definitely like to extend my heartfelt thanks to all of you for joining us today in the today evening, and of course to our wonderful speakers, Miss Snigdha, Mr. Yogesh Joshi, and our very own Dr. Ilamati Natarajan. Thank you, speakers. Thank you for the wonderful sessions. Thank you for guiding our students throughout. And I'm sure each session today brought in a wealth of practical knowledge, from understanding the foundations of NGS and multiomics to identifying the right tools and platforms, and finally to choosing impactful projects that elevate your CV and future opportunities. Uh, Okay, so yes, no, I have not forgotten. I remember the promise that I gave you all. Yes, so as promised, we do have a special freebie lined up just for our live attendees. I request our team to share the link in the comment. And it's something we have carefully designed to support your learning journey ahead. Please go ahead and download it. Link is there in the comment section. Okay, and with our so we very soon on 4th of August we are launching our NGS Multi-Omics data analysis course, and any kind of information you need, please feel free to dial our toll-free number, and we'll be more than happy to guide you through this process. Okay, and our project our course comes with 30 days project. You get to learn from the best in our industry, best experts that we have. Okay, the theoretical knowledge as well as enough practical demo sessions you will be having. It's completely online training session you will go through, and then you'll have to work, you get the opportunity to work on 2 months project work. And as Dr. Ilamati was suggesting, that if you work good and if some significant result comes out of it, we also support you in your publication. Okay, so you get to publish your data, you get to publish your work as well with us. So with let's wind up for tonight, and once again thank you for being part of this incredible learning space. Keep asking questions, keep building, and most importantly, keep believing in your potential. Thank you everyone, and have a good evening.